Hi everyone, my name is Dania. This is Lab 2, making dilutions for BIS3034 Biochemistry. In this video, I will explain my serial dilution experiment, which was assigned to me using a dilution factor of 7 times and a final volume of 70 ml per tube. So here is the template we were given. My stock solution concentration is 0.05 mg per ml of bromophenol blue. The assigned dilution factor is 7 times and the final volume in each tube is 70 ml. To calculate how much stock solution to pipette into the next tube, I use the dilution factor formula, which is dF equals to final volume over initial volume. So 7 equals to 70 over initial volume. So our initial volume is 70 over 7, which is 10 ml. So I need to pipette 10 ml from the previous tube into the next tube. Then I add distilled water to make a total of 70 ml. So how much would it add into the tube is 60 ml of water. Now I will calculate the final dilution for each tube. Tube 1 is 1 over 7 of the stock. One over 7. So the final dilution is 1 over 7 times. Meanwhile, tube 2 is 1 over 49 of the stock, which is 1 divided of 7 times 7. 1 over 49 times. And then the third tube, which is 1 over 343 of the stock. 1 divide 7 times 7 times 7 equals to 1 over 343 times. So after serial dilution, the color becomes lighter with each tube. So tube 1 is dark blue, which is least diluted, and tube 2 is medium blue, and tube 3 is a very light blue. This is because each step reduces the concentration by a factor of 7. The decrease is exponential, so the color fades quickly. And next is the final concentration in each tube. We calculated as concentration. The final concentration in each tube we calculated as concentration equals to stock concentration divided over dilution factor. So tube 1 is 0.05 divides by 7. So our final concentration is 0.00714 mg per ml. Tube 2 is 0.05 divides by 49 equals to 0.00102 mg per ml. And then tube 3 equals to 0.05 divides by 343 equals to 0.0000146 mg per ml. As you can see, the concentration drops very quickly. In exercise 3 of our lab, we measured absorbance at 590 nm. According to the Beer-Lambert law, lower concentration equals to lower absorbance. That is why tube 3 will have the smallest absorbance value. In summary, for a serial dilution with dF equals to 7 times and final volume equals to 70 ml, I pipette 10 ml from previous tube. I add 60 ml distilled water. My final dilution is 1/7 and 1/49, and lastly is 1/343. Color becomes lighter with each tube, and this matches the Beer-Lambert law. Thank you for watching.